Hey, welcome back to a special bonus module of Interconnected. I have, since I started doing this work, I've just been learning about geniuses left and right. And we got one on the call now. Shane Morris is a PhD in molecular biology and biochemistry, really up on the microbiome, really up on detox. And that's really the kind of cross section through which we want to interface here is, we understand the world is toxic. What can we do about it? And how can we use the microbiome as a friend or a friendly force in this? So welcome, Shane.
Thanks. Pleasure to be here. Yeah, you're so kind.
Yes. So let's kind of set the table here with detox going in, right? So the basics of detox. We talked about phase 1 and phase 2 a little bit in the series, and we touched on how dirty the world is and how challenging it is for us to do this. So what's your philosophy on detox in the microbiome? And let's jump right in.
Yeah, so the microbiome is, of course, I'm really passionate about the microbiome because we have a full lab dedicated to it. But as it relates to the detox, I have a really deep interest in two aspects of it. One is the health of the microbiome as it relates to food you take in. But in addition to that, we have to consider the waste products we produce that get shoved back into the microbiome. Right, and so you get this recirculation. So there's really two aspects to the toxic situation we have. Furthermore, we now know that the microbiome is intimately linked to the liver. I mean, there's a number of pathways, either via the endocrine system, the nervous system, and or the immune system. And because of that intimate relationship with the liver, we certainly know that the microbiome controls much of our detoxification fidelity as well as our metabolic system in the liver. So there's a lot of intricacies here.
So let's get into the depth of that. So there are interconnections between the microbiome and all of these detox pathways with the liver and all that. What does that mean? Let's kind of lay this out for the lay person. Is it sending information? Is it helping the detoxification process? Let's get into it.
Yeah, that's a good way to start because the microbiome is we can think of it as another tissue, right? It's a conglomerate of trillions and trillions of cells, and those cells contain trillions and trillions of genes. So the numbers, you hear this all the time, and I know it's a cliche, but we no longer have a 10-to-1 microbiome to human. It's about 1.3 to 1. The new data has come out. It's not as many cells we used to think it was, but there's still 100 times more genetic information, meaning that they make so many more molecules that interact with each other and with us. So let's start with that. There are so many things that they convert our food into, or there are so many things they convert other compounds into that communicate directly with our systems. Either they can be hormonal, they can be small molecules that interact with the immune system, or they can interact with the enteric cells. They can open up your tight junctions if your microbiome's not happy, and we clearly know that introduces toxins. They can communicate through something that we call the bile, right? We all have bile, and when bile dumps into the intestine and the colon, your microbiome can do one of two things. It can metabolize that bile into amazing compounds, what are called secondary bile acids, or primary bile acids. But if you have the incorrect microbiome, you can create essentially hormones from bile that target the liver and actually start turning genes on and off, exactly like a hormone. And that can raise things like fatty liver, right? Non-alcoholic fatty liver disease. It can also turn on genes that shut down cytochromes, so you no longer metabolize phase 1 and phase 2 properly. So the microbiome has a way to directly influence the way your phase 1 and phase 2 behave.
Oh, that's amazing. So let's get into kind of the plumbing here. Where does the bile dump into the intestines? What region are we talking about? And then let's talk about what these bile acids can do specifically.
Yeah, so if you're looking at, we're looking at the bile coming in up by the pancreas. It comes out of the liver, of course, the gallbladder, and it's up in the small intestine, right? Mm-hmm. So we're looking at the ileum, duodenum. Now, that part of the intestine that's not the highest density of microbiome, but is still an extremely dense microbiome. However, it does traverse the entire small intestine because its job is to help you sequester fat. It's to make fat bioavailable to us, right? That's one of its primary jobs. Another job is also to deliver waste ultimately into the colon and hopefully into the feces as it progresses through the entire upper and lower, small and large intestine.
To be clear, you're talking about the bile helps carry the waste through
Yeah,
as it goes, and are you saying that the bile has access to the small intestine multiple points, or dumps into the small intestine but then works its way down the path?
precisely. It's dumped in the small intestine to help you digest fats, but it has multiple uses, in fact. So one of its functions is to help us bind fat and take fat in as a nutrient, but it also delivers waste into your GI so that ultimately you will excrete it in the feces. There's some other things it does, and one of those is also delivering bile acids. These bile acids tend to become hormonal. They bind receptors in your body and they turn things on and off. Now, when things are working properly and you have a great microbiome, you create primary and secondary bile acids that do a body good. They help deliver information when they get reabsorbed, they deliver information to not only the enterocytes of your intestine, but also to the liver and to other tissues, mind you, to muscle, potentially to the brain. They act as hormones. When you have dysbiosis going on, when you have these organisms that convert bile into negative hormones that bind to receptors or antagonize a receptor, we create a problem. And that's where the microbiome, when it's healthy, it creates these bile acid hormones that are good for us. When it's not healthy, it creates negative imbalance in our hormone system.
Okay, so let's geolocate this a little bit, because we talked a lot in this series about SIBO and certain bacteria that don't belong in the small intestine. Maybe they moved down from the stomach or moved up from the large intestine, but basically you got bacteria that are in a region where they don't belong, and then you have overgrowth. You have methane producing bacteria and all that.
Right.
Does it tend to be the same kind of lineup of the usual suspects here? Like, is it the same kind of methanogens, the archaeobacters and all that have the same kind of deleterious effects with the bile acids, or are there other players there?
That's a great question. There are other players. So the methanobacters, they're interesting because you're right. They create these secondary metabolites from their own activities that can be dangerous, right? They can actually methylate things like mercury, which makes mercury more toxic. They can methylate other organic compounds, which makes them more, so let's say we're talking PCBs and other things, it makes them more available to the tissue to wreak havoc. But we have so many more players. There's about 1,000 species of microorganism in our body, and they fall under groups that the names you'll hear, things like Firmicutes, Bacteroides, Collinsella, allotypes. There's this huge diversity. However, when any one of these players come to play, and inside these species, there's subspecies, and many of those are what cause problem. Like, so you've heard of C. diff, Clostridium difficile. There's a lot of Clostridium species like Clostridium symbiosum that is a good guy. It actually helps you stay healthy and outcompete the difficile. But when you find a situation where, and this comes back to bile acids, there are certain bile acids that actually inhibit Clostridium difficile. But if you have a situation where you're producing a lot of bile and you have organisms that are converting it into the wrong kind of secondary bile, that allows Clostridium difficile to grow in your body. Mm-hmm. And now we have a problem. So as just a reference or as an analogy, that can exist in so many ways when a certain organism starts to predominate in the body, or it's no longer kept in check or in balance by other very healthy organisms. Because as you know, it's a dynamic system. With 1,000 species, some are growing, some aren't. They coat our entire GI tract, and so there's going to be this dynamic system going on where these millions of metabolites are being produced. And what we do is we have to, by the time we think about this, we have to create so many prebiotic situations along with probiotic situations and lifestyle and food that maintains this balance. And it can be tricky for some people.
Yeah, I mean, it's incredibly complicated, with all these kind of variables going around. And so how, like, when you look at this, and so you're trying to do some sort of microbiome based intervention, a lot of the docs that I've talked to will say, okay, look, you know what, let's go in, whether it's, a strong antibiotic or, some oregano oil or, black walnut husk or whatever it is. Let's beat down the colonies that are the aberrant ones that are kind of creating some of the problems, and then let's help re-inoculate and go. So what's your philosophy on that? I mean, it's just, I've heard a lot here, but I just want to see kind of what your strategy would be going in with someone, say, who has aberrant bacteria problems already.
Right.
When you go in.
Like some dysbiosis, which defines growth where it shouldn't be, or too much growth where it shouldn't be, or SIBO is another term, right, where small intestinal bacterial overgrowth. So let's begin with the idea that there's two types. You can have pathogenic scenario where the organisms that are growing and taking a hold in your body are dangerous because they're producing pathogenic substances, right? They're toxic. You also have a situation where regular bacteria that tend to inhabit you in a commensal way, where they're not really a problem until they start growing. And as they start to grow out of control, it's not that they're producing a toxin, it's that they're infringing in areas they shouldn't be, or they're creating tight junction issues and so on. In both cases, right now, based on anecdotal and very little studies, I'm big on knocking them down. I prefer the natural, what we call the natural killers. Like you say, there's oregano, there's a number of other herbs, and they're usually a high terpene or high essential oil herbs to try to beat down some of those organisms. You can also throw in their intermittent fasting to help drive that down, so a little starvation. Bacteria respond quickly to starvation. 3 days and you start to lose a lot of your bacteria. Now there's this core set of bacteria that exist in each of us that will recover. But in the meantime, hopefully the ones that shouldn't be there don't come back as easily. So we have this starvation, we have a kill philosophy, but then as we bring them back, I have a very distinct protocol that I try to use. And that is, one, get back into the prebiotics quickly. And when I mean prebiotics, I'm not just talking oligosaccharides, I'm talking about using really colorful plant compounds, meaning the polyphenolics, the flavonoids, the genistein, berberine, and all these amazing compounds that we normally think of as therapeutic herbs. They tend to be really amazing for the microbiome. We used to think of them for us. Well, I would argue that some of these herbs are better for your microbiome than they are you. That's where I go with this. And then you introduce probiotics subsequently, because probiotics, they may be transient or they may want to live in you. But one thing about the probiotics we grow is they produce things like bacteriocins, and bacteriocins are nature's antibiotics. So they can help knock things down. But you just have to temper it, just depending on the person, because you don't want to trigger any other growth. So you bring it in slowly, starting with prebiotics and then move into the pro.
Let's jump back into this intermittent fasting conversation real quickly, because starving them out means you're starving everyone out. So how do we know that the good guys are outlasting the bad guys? And at what point do we know that that's actually a winning strategy?
Well, I can tell you that some of the few studies, and again, these are very minimal studies, and the work we've done in our lab shows that the organisms that we have an intimate relationship with us, they find a way to survive. So when you think of your intestinal tract, both the small and large, it's full of nooks and crannies, and you have the mucosa, and you have the body's actually producing food for the microbiome, believe it or not. So things like chondroitin and mucin and these other glucosaminoglycans, these are actually food for them when there's no food coming in. When you're not eating prebiotics, you feed them. So these guys—
How? Sorry, real quickly, like you're producing the chondroitins and all this, how are you feeding them into the GI system?
They're secreted by the enteric cells. They're secreted by the cells that line your intestine and your colon. They secrete these polysaccharides into the colon and the small intestine to feed organisms.
Interesting.
Yeah.
You're the first expert who's even spoken of that in
Oh,
this
yeah.
thing. And so we actually have, not only are we feeding this way, but we will feed from our own system through the enteric cells into the GI tract.
That's right.
Entice the right organ. And at that point, then that's the food for the right bugs because those are the symbiotes that we're feeding very intentionally.
Exactly. Yep. So you have mechanisms to, hopefully when you're healthy, you have mechanisms to stabilize and maintain a healthy microbiome. Of course, as you and I know, when you have chronic inflammation, toxins, etc., that switches everything, right? That creates an opportunity for the non-commensal, sometimes pathogenic, to take hold. And in the case of like H. pylori, when that mucus thins too much, something like H. pylori, which can be a commensal for many of us, will start to get closer and closer to your tissue and can create a problem when you're not feeding it properly, when you're not maintaining this terrain, so to speak. The body's terrain is designed for the microbiome. It's a beautiful interplay when you really get down to it.
Oh man, that's for sure. I mean, this is one of the most marvelous things I've ever encountered in my career. I mean, just learning about this, it's blowing the doors open on everything we know, right?
I agree.
It's really something. So, all right, let's just stay on this concept here. You have intermittent fasting, so you take 3 days of water fasting. You slough off a lot of the bad guys. Your body starts feeding the good guys. Then you come back with polyphenols, berberines. You come back maybe with inulin, some things like that. Lots of colorful vegetables, supplements if you need them, and then you start to slowly kind of rebuild and re-inoculate as you go. What would like a reasonable protocol there be for someone? I mean, I know it's really personalized, but are we talking about lots of prebiotics early on, or do you phase them in so that it doesn't hit the system too hard?
I would say that you phase them in, and the reason for that is certainly the microorganisms are going to be extremely happy to see these prebiotics coming in, especially the colorful ones. And however, and keep in mind, it tends to be when I say, I'm gonna have to define things. So the polyphenolics and the flavonoids and the alkaloids like the berberine and hydrogen, those things don't tend to create an issue. They tend to actually be somewhat healing as you introduce those. So I think you can introduce those immediately. The ones that are a little bit trickier are things like inulin, the fructooligosaccharides. Sometimes you need to switch to galacto-oligosaccharides, because the oligosaccharides, depending on your microbiome and where it's at in the turnaround healing process, can metabolize those quickly. And in many instances, they turn them into gases. And so there can be, you find that some people will get into a little bloaty or crampy. So phase those in a little more slowly and try to be diverse. Don't hit them strictly with a fructooligosaccharides and ignore the other oligosaccharides, right? And that works with the food too. When you bring in a little diversity in the carbohydrate realm, complex carbs, you'll notice that there's a sharing, because the other thing that we need to briefly mention is there's a hierarchy, or there's this beautiful chain of command that also happens in the gut, especially with your own microbiome, is the first, an oligosaccharide will be encountered by a Firmicutes, let's say. That Firmicutes will turn that oligosaccharide into sugars. So its waste product is sugars. Then the next guy down grabs those sugars and converts those into things like butyrates or propionates or other compounds that then feed the third level of players. They take those butyrates and turn them into energy or turn them into another metabolite. So there's this beautiful interplay between your microbiome. And ultimately, not only are they helping each other in this community, they're secreting compounds into your bloodstream, through the villi as well as colon sites, and they're feeding you a number of things like serotonin, for example, or other, in stimulating dopamine or stimulating the neuroendocrine system. So there's a really amazing sequence of events, and it's all metabolic. Keep in mind that it's not just that the microbiomes are there, they're turning all these things that you're eating into secondary, tertiary metabolites that then trigger your own genetics in some cases.
Amazing. So when you have someone who's addicted, shall we say, to sugar or refined carbs, and we know that that's kind of feeding the wrong guys, and we back up to this understanding that you just laid out, where you have this sequence, you get the first at bat that will take certain complex carbs and break them down into sugars to feed the next at bat, which will then produce the butyrates and all that. Is that where we also, because I've heard some through our interviews of, there's actually energy metabolism, right? Like we can actually get energy. There's more ATP being produced by, eating the right foods type of thing. So how does that feed? So it's feeding the bacteria that are eventually feeding us, but is it also shifting our caloric intake or input? Like, are we, if you have the right microbiome, what does that do to the way you metabolize and like kind of draw energy from these complex carbs?
Yeah, that's a great question. That kind of leads back to the beginning of our talk too. So let's just take glucose, as a primary, and fructose. So glucose, most organisms in your body can handle glucose extremely well. But the thing that happens if we're eating a lot of sugar, it drives a group of organisms that love sugar more than others. And so what you're doing is you're creating a competitive edge for an organism, whether he be a good guy or a bad guy. You're creating this competitive edge when you're just eating glucose, and you're losing all those organisms that need the complex carbs. So they're shrinking in volume. The glucose lovers are growing in volume, and they don't necessarily now start feeding the butyrate producers. And so you have this, it's a form of dysbiosis. That's one way. Now you get this glucose production, and because of that, you're not only altering the microbiome, you're altering the signals that your body is sensing from the microbiome, right? So your body's listening to the microbiome constantly through three paths. One is your vagal nerve, right? So the brain, the reason the vagal nerve exists, in my opinion, is just to stay in touch with the microbiome. I believe that's its primary function. Signals coming from the microbiome are going directly to the center of your brain. Secondly is the largest immune system, the GALT, exists in the GI, right? Gut-associated lymphoid tissue. That's your largest piece of immune. And finally, your endocrine system communicates. So those three things happen. Glucose disrupts that, as we know, in a number of ways. Now, insulin becomes an issue, and so on. We start getting a lot of sugar and then a chronic. That's one example. I want to point out another one, which takes us back to liver, and that is the fructose issue. So fructose tends to increase what we call gram negatives that produce lipopolysaccharide. Have you heard of LPS in your discussions?
Oh, yeah.
That's a molecule on the surface of gram negative organisms that can cause issues with the liver. It's a toxin, essentially. So when you get fructose, a lot of high fructose corn syrup, you remember when that was the craze? And everybody's like, well, it's great. It doesn't raise your insulin. What a great idea. No, it was the dumbest idea on the planet, because not only is the fructose changing your microbiome, it's sending now signals. That microbiome that's living on fructose is sending signals to the liver, and it's those signals via bile and other molecules are telling the liver to convert fructose into fat. So that's what drives the non-fatty liver disease, non-alcohol fatty liver. Mm-hmm. And we also know that drives, that's one of the etiologies of type 2 diabetes. And so we start creating these connections to these chronic issues. And one of the major players is our microbiome, without a doubt.
It's a lot of people get freaked out over this because you use the word fructose and they say, well, a lot of these other experts are saying you need to eat a lot of diverse, colorful fruits and vegetables, right? Yeah. And so let's talk about fruits versus high fructose corn syrup, and let's talk about what's too much, basically.
Yeah, it's a great point. I mean, I love fruit, and we know the planet loves fruit. I mean, if we go back to the oldest cultures we can find, and especially in equatorial regions of our planet, they subsisted on fruit all the time. When there wasn't meat available or something, fat, it was fruit. We know that. Trees were plentiful. I mean, that's just the way it is. And it wasn't necessarily detrimental. Why? Because it was part of the complex of other plant-based molecules like the polyphenolics and the essential oils, by the way. When you eat a citrus fruit and you're eating it, you're not only getting the bioflavonoids, polyphenolics, you're getting carotenoids. Anything yellow, orange, red are carotenoids. In combination, these things drive a diverse microbiome, which leaves the fructose to be metabolized efficiently by the microbiome into secondary metabolites that are great for us. When it's fructose corn syrup, that fructose bypasses or essentially floods the microbiome. The guys that love it will start to grow out of control, but the rest of it just penetrates our body, just heads directly into the bloodstream.
Fascinating. So, a lot of the arguments I heard about this is like, look, you can't get enough fructose because, I mean, how many apples can you eat if you're going to eat whole food
Yeah,
or fruits? It's just there's a rate limiting factor with, satiety and space and water and all that. But what I'm hearing here is the carotenoids will come in as a secondary player and support the right microbiota that would then help with the breakdown of said fructose so it doesn't become hepatotoxic.
exactly. Because remember, when you're feeding your microbiome and it's growing, these cells double every 90 minutes. So if you have a million cells to right now, between 30 and 90 minutes, depending on the organism, you now have 2 million, 4 million, 8 million. So, that's a dynamic that requires a ton of energy, a ton of primary sugars. And if you have a healthy one, it's going to deplete your diet of a lot of these nutrients and make other nutrients for you, right? You're feeding your microbiome the fructose and the sucrose from plants. It's going to take its share off the top, which really prevents that hepatotoxicity because it's using those to create a lot of other things with the help of these other nutrients. I've found in the lab, by the way, we grow a lot of unique organisms in our research lab. We're growing human organisms that we consider probiotic. It's not there yet. We're going to, be introducing them sometime in the future like others, but they're unique. And what we're finding is they need these essential nutrients. They need the carotenoids. They need the riboflavin. They need all these essential compounds for me to grow them, which tells me that when you eat these things, you're not just eating them for you, you're actually eating them for the microbiome. And then the microbiome will give you back what's left or what you need on top of their needs.
Amazing. So some of the most amazing stuff that I've learned through this is, so there's all these innovations in, like, say, postbiotics, where there are particular bacteria found only in the vaginal canal that secrete a biofilm that has a very strong antibiotic function that basically crowds out any invaders.
Mm-hmm.
And it's a human-derived bacteria, and it's very local and targeted. So let's figure out how to use that for vaginal tract, or infections. And so the idea of not having to take a 10-day amoxicillin cycle every time something goes wrong and having very targeted postbiotic therapy is really interesting. So I'm curious what you're doing in there. I mean, I don't know if some of it's secret. Just tell me what you can. Like, what are you guys working on?
Well, we have I think I have at last count, I've got about 15 unique organisms. One of them is hitting the news. You've heard of Akkermansia muciniphila, maybe? That's called the skinny bug. Well, that's one that when you take it out of a skinny mouse, if you just take Akkermansia and place it into an obese mouse, same diet, same everything else, it loses weight. That organism drives weight metabolism or metabolism. You can also see that bug. It's essential to the cold heating cycles. Like, if you enjoy jumping into a cold ice bath and then you jump into a sauna and back and forth, Akkermansia actually comes into play during those pieces. It's really fascinating science. That's one of them. The other ones we have is, you're right, there are some we have, like, for example, Staph epidermidis. That's like your vaginal one. So Staph epidermidis is a staph. You've heard of Staph aureus or MRSA. That's a scary one, right? MRSA is the scary methicillin-resistant Staph aureus. Staph epidermidis produces compounds, again, they're called bacteriocins, that if you have a healthy Staph epidermidis on your skin, then you are unlikely to ever get a staph infection, because epidermidis is competing away the aureus. So those people with beautiful skin have never had a single bit of acne or lesions. They have a really healthy microbiome that produces these natural antibiotics. And it does it in concert with your own immune system. So it's a really brilliant, again, it's a brilliant thing. The immune system feeds these organisms by the sebum. So, we produce sebum or hydrolipid layers. Half of that goes to feeding your skin microbiome, no different than when you feed orally or when you produce mucins to feed the GI organisms. So you're right, there's these dynamics that we have that we absolutely want to maintain. And if we maintain them and we don't use toxic antibiotics or alcohol wipes or all these other things that we do to destroy everything, then when we encourage the growth of these good bugs, they will create these metabolites that benefit us in a number of ways. Yeah.
Let's talk about the complexity of humans. So, there's all this excitement about acromantia with mice, right? It's like, oh my
Right.
God, we found the skinny
Yeah.
bug. Everyone eat this thing, and weight loss is done. That industry just comes to a screeching halt. And we realize it was more complicated than that. So let's talk about the complexity of that and what you're wrestling with to actually, make that work. And to me, I think that the spoiler alert would be it has to do with the last 20 minutes of what we talked about and how there's stages and sequences in what the human microbiome needs to receive and do in kind of some of these, like, it's like a bacteria A produces substance B, substance B is eaten by bacteria C, and so on and so forth. So
Right.
I'd love to hear this played out because everyone wants it to be so simple. It's not.
No, it certainly isn't. And as complex as mice are, we are, order of magnitude more complex. And not only that, when you think of animal diets, they're quite narrow, and humans are quite broad. And so, in fact, we've really developed this breadth of dietary intake that drives not only the diversity, but it makes these players impact part of a team or part of a community. So you might be a good Akkermansia carrier, but you still struggle with metabolic issues because of the other players or the other genetics, right? So we know now that I've mentioned, the interplay between our microbiome and our immune system and our GI and our, neurological and so on. Well, there's genetics that at play here too, where because of where you come from, where are your ancestors equatorial? Are they northern? There's this genetic piece that drives these relationships in many cases. And of course, diet can influence it. Certainly when you change your diet, you can start to change these things. But there is a genetic component that we're just beginning to understand. And we have to understand that. We have to understand the microbiome in further depth in order to say, okay, can Akkermansia help some people? Absolutely. We're going to find these people that improve their Akkermansia levels, and they do the microbiome test, and they show that they went from 1% Akkermansia to 2% through supplementation of Akkermansia, and their metabolism is improved. Brilliant. It's going to happen. But there's going to be other people that already have 3% and they're struggling metabolically. It's no longer Akkermansia. It's probably Christenseliniaceae or another combination of organisms that have a role in their world to make that metabolic issue good. So will we start teasing these out? We will. And we start with things like the vaginal tract, right? We're seeing this interplay between estrogen and these organisms. Like I have one that we're growing it's called Lactobacillus vaginalis. Well, of course, you can imagine where that's from. It's a vaginalis one, and it does produce a natural antibiotic. But it also seems to be in higher concentrations in childbearing women or premenopausal women. When they go through the change, when they go through perimenopause and menopause, the hormonal drops actually create a situation where vaginalis declines in growth. And we don't know what that is, but what they found is that they introduce estrogen just into the vaginal area. Those organisms recover and flourish. So there's this really amazing dynamic between not just our diet, but other things like hormones in the case of women's urogenital tract. So there's these amazing things. You're right. As we learn, we're going to get better. In the meantime, I'm still a firm believer as a researcher and of course as a proponent of wanting to get prebiotics and probiotics out there. I think it's worthwhile to try these or to work with these organisms in humans because there's very little downside that I've discovered. I mean, if I try a vaginalis on a woman and it doesn't take root and work, then we have to go somewhere else. But when we try and it works, that's a good thing. We've given them a benefit. With the rest of them, we have to keep going. We have to keep trying to get better at it. Yeah.
Man, it's such an exciting field. And I know we kind of danced around a lot of places because you're just into a lot of interesting things. So I'm going to kind of bring us back in our closing minutes here to detox. How does one think about detoxification with the microbiome? What do you do? Like, what would be the right way to think about saying, okay, look, I live in a toxic world. down there, they're spraying glyphosate. There's a diesel truck that just drove by. I haven't had the cleanest diet my whole life. Help me, I'm a sinner, right? Like it's really
Right.
hard because we feel like we're too far gone. How do we fix that?
That's a great point. And that's really come to light recently with looking at the fasting element. So for example, we know we're toxic. Let's say I'm toxic and I know, I've got years of it, months of it, whatever it be. I'm living in a toxic world. So how do I reinvigorate like my own body, like I would? How do I do that to my microbiome? A lot of the same tools apply. So first of all, we want to supply really good nutrients in the form of probiotics, rich in color, rich in vitamins. Supplement if necessary. I'm still a believer in that. Supplement with the trace minerals and the vitamins that just aren't abundant in some of our foods. Secondly, do these what we call intermittent fasting or diet variations. Drive our bodies in and out of these states, because we know that by doing that, the microbiome is rhythmic, just like we are. We have a circadian rhythm. Women and men have a rhythm monthly, right? Women clearly have a monthly rhythm. So do men. Get your microbiome into a rhythm, not only with diet, but in other ways. Get it into a rhythm, making sure that your sleep cycles are there, making sure that every once in a while when you're eating, eat things that cleanse. That tends to be supplements, but things like carbons and fulvates and lignite, things that they go through and they collect not only dead bacteria, but they collect toxins that can persist, right? And there's two ways we want to do that. One, you're collecting toxins that you're ingesting or you're being exposed to in your lungs and your water, et cetera. But you're also collecting toxins that are a result of your liver's metabolism, right? Phase 3, that's the excretion. Phase 1, phase 2, but then phase 3 pushes it into excretion. So make sure you're getting that off of you and out of you. Skin, vaginal, urogenital, and even lungs, right? Make sure at some point in time you're supporting the lungs through various aspects of not only, of course, trying to breathe well, but there are a lot of herbal and other nutrients that we know work for the lungs. So in that sense, I'm certainly giving you some, I have a bias towards a little bit of supplementation, clearly, prebiotics as well as diet. But in each one of those cases, when we try to detox our body, what I'm saying is detox your microbiome in the same way. They are just another critical organ. They are your brain. They're like your brain. They're like your liver. They're like your heart. You can't live without them, and they need the same love and care that you would want to give each of those organs. And they indeed are an organ.
Yeah. Now that we've kind of found this, the question is, it almost seems like they're a super organ and they have much more to do with every other organ than we ever thought, right? Talk about, being interconnected, but I think this is just the beginning. We're just scratching the surface of what
we are. Yeah, you're absolutely right. This is changing the way we think about health, the model of health. And I think it's going to change medicine too, because we have an opportunity. This is the focus of the immune system. It's the focus of the GI health. It's the focus of the gut-brain connection. You're right. The microbiome is central because of its diverse genetic. It gives us 100 times more genetic power than our own cells do. This is just the tip of the iceberg. I've seen entire immunology departments switch to microbiome research in universities because they realize the immune system is nothing without the microbiome. The immune system doesn't learn, it doesn't adapt, it doesn't do what it's supposed to do without the microbiome. In fact, it goes haywire.
It's amazing. I've been, it's funny, you get your occasional kind of, how do I say this, propaganda doc who's like, there's not enough research, right? And so there's these kind of old crusty grumpy guys that are usually like poo-pooing the microbiome, saying we don't know enough. And I've been to Caltech, I've been to Johns Hopkins, I've been to Stanford, I've been to the top research universities where entire floors, if not multiple floors, are now dedicated to studying the microbiome with the smartest people in the world, just going like as fast and furious as they can. So the answer there is, this is real. This is happening. This is revolutionary. And don't listen to the crusty old guys that are, part of the old guard.
That's right. And it's such an amazing, the microbiome is in our control. It's in our health control. It is us. We can take control of our health this way. And I would tell all of your listeners that although we don't have all the answers yet, and the science will continue to push out amazing new data. That doesn't mean we can't start now with dealing with what we have now and use it. Use our prebiotics, use our nutrients, use our plants, use our foods as the tools. And if we don't have the answers at the moment, you know what? Our industry needs to push forward in doing it so that we capture this health dynamic or this health paradigm. I don't want this health paradigm to go to another industry. I want those of us that care about our health and the health of us, the health of the planet, the health of our animals, they're all connected to the microbiome. We need to take advantage of it so that it doesn't become this crusty old science.
Yeah. Yeah. Well, it's just, the commercial interests are not interested in people actually being well. The money's in sickness.
Yeah,
so
exactly. It's exactly right.
Kind of dark.
Yeah.
So, man, I really appreciate what you're doing over there. I can't wait to, kind of dig in more and figure out, what you're working on in that lab, and we'll get there. We'll share it with our audience as things start to kind of come forth. Dr. Shane Morris, thank you so much for your time.
Yeah, thank you.